of today 20th lecture series on of Indian Phytopathological Society, Platinum Jubilee on the occasion of 75th years Azadi Ka Amrit Mahotsav, organized by our most popular plant pathological society, that is Indian Phytopathological Society of India, New Delhi. First of all, I would like to thank our speaker, today's speaker, Dr. Bilal Ahmad Padler, Associate Professor, Division of Plant Pathology, Sere Kashmir, University of Agriculture Science and Technology, Jammu and Kashmir. Thank you very much, sir, for accepting our invitations. Lecture on Fragilis vulgaris, Coletotrichum lindemethynum interface inside the futuristics roadmap. Today on the 22nd February 2022. Now I request to all participants to like, ask question in comment box and subscribe the lecture series in YouTube channel or in ongoing lecture series. These all lectures are also available in our Indian Phytopathological Society website. And then now I request to Dr. Ganesh Prakasa, Scientist Plant Pathology, Division of Plant Pathology, IRI New Delhi, to welcome our speaker, Dr. Ganesha.
Yeah, very good morning to all of you. It's my privilege to welcome the speaker on behalf of Indian Phytopathological Society and my own behalf for this 75th Indian Phytopathological Society Platinum Jubilee lecture series. Today is 23rd lecture and the speaker is Dr. Bilal Ahmad Badar. He is an assistant professor, Division of Plant Pathology from Scouts at Kashmir. And he will be delivering lecture on Casualis vulgaris, Coletotrichum lindemethynum interface, insights and futuristic roadmap. I cordially welcome the speaker also. And it is undoubtable that this lecture series, last 3 months lecture series, is already created a platform for all those who are here from across the country to share and inculcate the information and to recommend the solution for the sustainable management of plant diseases that affecting our crop system, thereby helping the mankind of managing the crop diseases. Today's lecture also definitely will help us to know about this important national plant pathogen and its management. I would like to brief about the speaker, Dr. Bilal Ahmad Badar. I already told he's an associate professor from Division of Plant Pathology, Scouts at Kashmir. And he got many awards and honors and publications also. More than 50 publications he is having, and he got a lot of best paper and best oral presentations from different national and international symposiums. And he is a Indian National Science Academy INSA fellow during 2014, and he got a Raman fellowship in USA for 1 year by UGC government of India during the period of 2014 and 15. And he has awarded appreciation from Vice Chancellor Scouset for getting Raman fellowship, and he got a project proposal and reviewer from the Department of Science and Technology. His main research area is he's working on bean and the anthrogonous interaction. That's why we aptly selected the speaker for this today's lecture series. I cordially welcome the speaker to initiate his lecture. Thank you very much.
Thank you, Dr. Gupta and Dr. Ganeshan, for giving an introduction. First of all, I would like to thank Dr. Pratibha Sharma, the President of Indian Phytopathological Society, other officials of Indian Phytopathological Society, scientists and the students. A very good morning to all of you. Before going to deliver my talk, I thank the IPS for giving me an opportunity to say about the futuristic and current insights on the host pathogen interaction, which I have chosen that Phaseolus vulgaris Culturetrichum linemethianum, that means common bean, and it is anthracnose pathogen. Now, I have divided my talk into 5 categories, 5 broad areas. One will be the bean anthracnose races. As we know that Culturetrichum are races available present in the world as well as in India also. So I'll be talking on this. Then I'll be moving to genetic diversity using molecular markers. Then my talk will be on pathological gene discovery, pathogenicity genes. We know that the pathogens have in their genomes armory, which they express during the colonization and infection process. These are very, very important, and we must understand the pathogenicity genes so that we could target those for the management and understand the dialogue very well. Then I'll be coming to resistance genes, how these are structured in the beans and what insights we have about this. Then ultimately I'll be coming to the futuristic roadmap, where I'll be telling that how this recent advances can help us in understanding this dialogue, and so that we could manage this disease in a better way. And since the common beans, most of these common beans are grown by northern India by small farmers, and skyrocket costs of inputs like fertilizers, pesticides, they are very, very costly, and they increase the burden to the farming community. So understanding this anthracnose resistance and imparting those genes into our commercial varieties will be a very valuable and eco-friendly management capsule for our farming community. Now, coming to the bean, we know that common bean is one of the important crop of northwestern Himalayan region, and India is the second largest producers of common bean in the world after Brazil. And in northern India, we have a cousin like Rajma Chawal, very, very famous in northern India, and it must be, in a week we use it, and it is a low glycemic index crop, legume crop with high proteinaceous value, and it has a proteinaceous power. And this interaction started when Barris, he was a student, BS student in Cornell University, and his teacher told him that you collect these isolates, then inoculate the Culturetrichum linemethianum to varieties on this. He collected diverse isolates when he inoculated them to the beans. To his surprise, he found that some of the varieties were resistant to some isolates, but same varieties were susceptible, which later on Floor gave a gene for gene hypothesis later on. But he, in his paper, gave the differential varieties, but then it became Stackman's differential set, differential lines from that. But Barris was the first who first time identified this phenomena. Now, both beans as well as this pathogen is highly diverse. Neither the pathogen nor its host is native to India. We have two major gene pools of common beans. We call them as Andean and Mesoamerican in the United States. It has moved to Europe, and from Europe it reached to the India. And still, not being a native, we have highly diverse nature of both common beans as well as its pathogen. And in India, nearly 45 races are reported, and in world, nearly 182 races of this pathogen are reported worldwide. Now, coming to the genes, there are 40 resistant genes which have been identified, and most of these genes have been identified in US, France, and Brazil. India has only done the resistance evaluation, but we have made some inroads in that. Now, bean genome was cracked in 2014, and the 632 MB genome has paved the way to not only to find map economic traits, but we have now find mapped many of the resistance genes. Among these 40 are now find mapped. We know the positions of these genes and their candidates as well. And this has also helped in cloning of the first common bean gene bean anthracnose resistance gene also recently in 2021. Now in 2015, another inroad was done when a bean chip with 6,000 K bean chip transcripts was exploited, and it's being exploited worldwide to understand this bean anthracnose as well as other host pathogen interaction dialogues, also for economic trait identification. Then we know that plant molecular biology has many advances, and it has also touched the field of plant pathology. So next generation sequencing has been used to understand this dialogue also. I'll be talking about this. In 2017, a Canadian group, DCORS, for the first time decoded the genome of Cultotrichum linoleum, the bean anthracnose pathogen. But after mining, he later on mined it again and identified nearly 200 effector genes, but these are only bioinformatically validated. They have not been validated by wet lab. And it has paved the way how this pathogen has biotropic, nectotropic switch, which genes are expressing in the biotropic phase than in nectotropic phase, and between biotropic and nectotropic phase. It's very, very less studies have been conducted on that system. Now, coming to the first symptoms, now this pathogen affects all the above ground parts of the plant, right from the seed, and as soon as the most conspicuous symptom which appears on the bean pods is the rusty brown pustule. Here, it's sunken, surrounded by the brown or reddish margin with abundant acervillus in it. And if the infection is very, very high, the market class of these dry beans become very, very limited. They are not being marketed very well because they will not fetch high prices because of not having the good quality. There will be dots, that anthracnose dots on it. And if these seeds will be planted, the crop will get infection early, and there will be a yield loss of more than 100% sometimes. And in leaves, it produces characteristic symptoms of blackening of the veins on lower side, and it is how much devastating this disease may be in northwestern Himalayan region. Now, coming to the races, as I told you that it's a hemibiotropic pathogen having a gene for gene interaction with the common bean. And as I told you that Barris was the first time used the differential varieties phenomena, and Drishfraut and Davis later on gave an international differential set. But this differential set was later on, the Pasteur corals remodified it, and there are 12 differential varieties. These are listed here. And we have gene pools, as I told you that there are 2 major gene pools. We have 4 varieties in it, which are of Indian gene pool, and rest of them belong to Mesoamerican. Their genes are well decoded here. And the Pasteur corals for the first time gave the nomenclature, that is binary value. And when we give binary values, for instance, how we designate race 73. Now, an isolate is susceptible to Mexico 222, Cornell, and Michelide. If we add up these, it comes 73. So we classify this isolate as a race 73. And as per this classification, there are races from 0 to 3993, up to this much. And if we add up all these, we will come up to 4095. And in world, as well as in India, there is not a single isolate which has the ability to overcome all these resistance genes and reach to 4095. So we have some resistant genes which can be very effective in managing this disease. Now coming to as I told you that there are nearly 182 races reported worldwide, and these races are different. And if we come to this risk nomenclature, there are particular races which are prevalent to a particular region, and they are very, very more concentrated efforts are being devoted towards this. For instance, if we say to the Brazil, race 65 is very, very dominant race. And most of the breeding approaches in there are devoted towards this race. Rest, their frequency is very less. As far as India is concerned, we are concerned. Now, race 513 is well distributed, but race 503 is distributed on all the northwestern Himalayan region states. I'll be coming to later on. Now, If we come to the bean anthracnose research in India, as far as this research is concerned, it's mainly concentrated in Himachal Pradesh, Krishi Vishwavidyalaya, HPKV, CSK, HPKV, Palanpur. Then Scotch J&K is doing a lot of work on common beans. There are some exceptions, like GB Pant University, there it's have done the research. Similarly, because this disease was first reported from Nilgiri Hills, none other than Butler. So US Bangalore has also worked on this interaction a little bit. Now, if we come to the interaction, this race interaction, now we have classified in India, there are 4 types of 4-class interactions on this. Now, type 1 class is that we don't have any isolate which is able to overcome all the resistance genes. It's classified as race 0. So we have few races which are able to infect only Mesoamerican cultivars. We have a few isolates which are only able to infect the Indian cultivars. But majority of our interaction, what we have observed is that we have the isolates which have the ability to overcome both Indian as well as the Mesoamerican gene pool. Now, this is very, very unique. We are unique in this. Now, it's why? Because if we come to the US, Indian genotypes, Indian Cultrotrachum lineamethyanum isolates are always able to overcome Indian genotypes only. And there they have their breeding approach is that they incorporate introgress Mesoamerican and Indian gene pool together, and that gives them a very, very good control measures. But our race spectrum shows that our race spectrum is totally different. And we have to have a different strategy among these differential cultivars which contain these resistance genes. Now we are having 2 varieties for which we don't have any isolate which can overcome this resistance gene. Among this, now this AB136, I'll be coming later on, it succumbed to an isolate, but we have still one variety like G233. Very important, it contains 3 genes in its background, but the problem with this cultivar is that it doesn't flower in Indian conditions. Why it doesn't flower? Because it's a short day plant and we have a long day periods in Indian conditions. So we face a lot of problem in getting the flower and introgressing these lines into the cultivar. But now coming to this is our recent race spectrum. We revisited this host pathogens spectrum recently, and we collected isolates from all the regions, and we identified race 503, a unique race to all the states, and there are different races, and this 139, one single isolate. Now this race AB136, which contains CO6 and CO8, a recessive anthracnose resistance, it got succumbed to the pathogen nearly after 20 years. We started this work 20 years back, and it recently succumbed to the, it shows that the pathogen is evolving and new races were identified from the hotspots. So this gives us an indication that this pathogen is evolving, and we have to monitor on regular basis this race spectrum of northwestern Himalayan region, so that if we breed a variety, we should have with us a repository that we could deploy another variety with same agronomic traits within a short period of time. Now, since I told you that neither the pathogen nor its host is native to, still we are having high diversity of this pathogen. It gives us an indication that something should be, there should be some mechanism behind it. So we started recently working on it and identified and crossed spatio-temporally diverse, like isolates from Himachal Pradesh and Jammu and Kashmir, and their space and time right from 2003 to 2021, we used to cross them together in multiple combinations, but we only find Protoperithecea, but it was without the Ascus and Ascospores. And it led us to that most of the 95% of interactions were negative. That means they couldn't produce, only Acervulus was present, and only 5% were potential that were not productive. So this gives an indication that we don't have the MAT, because this sexual reproduction in Cultrotrachum lineamethyanum is governed by the 2 genes, like MAT1-1 and MAT1-2. But in our isolates, we only identified MAT1-2. We couldn't identify MAT1-1 in our studies. So this sexual reproduction is not going to increase the variability in this. Then we started looking at whether the heterokaryosis has any impact or vegetative incompatibility has any role in generating this huge diversity what we have in bean anthracnose bean pathogen. So we identified that yes, there are vegetative heterokaryosis, vegetative incompatibility is happening, and most of the categories, and it was interesting to identify for the first time NIT5, NIT6 interaction, which has not been yet observed in culture trichome. So this is our studies, which shows that yes, really we have heterokaryosis, one of the mechanism of resist variability in the pathogen. Now coming to the genetic diversity, most of the diversity studies in bean anthracnose have been carried out using the dominant markers like RAPD, AFLP, ISSRs, all these around the world, not only here in India, around the world. And most of these studies also have shown that the pathogen is highly diverse in nature. Now, these are some of the results we identified, a lot of diversity across these 91 isolates of culture trichome minimethionum using RAPD. We did population biology studies also, and we found that there are, even though the pathogen is highly diverse, but when we come to the population biology studies, we see that there is a clear-cut differentiation happening in the population. That means the farmers are using their own seeds, and there is not a gene flow happening in this pathosystem in northwestern Himalayan region. Now, we had speculated that there are some missing links in our evolutionary model, which we cracked in 2009. But a recent study has shown that yes, these it's evolving. The pathogen is evolving on different fronts, both at molecular level as well as at virulence level. So we have remodeled it in it. Now, as I told you that there is no neutral marker system, SSR marker system was not available for this pathogen over a long period of time. Now, 2017 genome was available publicly, and we took the advantage of that genome, and we mined some of the SSRs, nearly 19,000 SSR markers for this. We took different culture trichome species at that time. These belong to the different clades because there are different 14 clades in culture trichome on the basis of molecular taxonomy. So we took those clades also and identified a differential set in this, and this is our preliminary results, which shows, and this is how the genomes were available. We fed, and these are some of the top 20 SSR markers in this, then these motifs, then their validation and clustering. Now, earlier clustering had not categorized our isolates onto the host geography, but when we used our neutral SSR marker set, we could easily, although the isolates numbers are very less, but we found some of the host specific geographic orientation of these isolates based on these neutral SSR markers. Now, as far as pathogenicity is concerned, in worldwide, only 5 pathogenicity genes have till date been decoded in culture trichome lineamethionum. Now, since the genome was available, its repository was also available. So this REMI has got many disadvantages. So we took an advanced agrobacterium mediated transformation so that we should have a transformation protocol with us so that we could explore it and use the site-directed mutagenesis approach to identify these pathogenicity genes in the pathogen. And then using those classical experiments, we could give a function to each gene. And we got a very good success in optimizing this protocol. And this is our some results from that. And we also, since we had nearly 2,400 mutants, and it was very, very difficult to handle those mutants and then use the classical, which is that you have to germinate the seeds, inoculate them at this level and incubate them. If we use this type of technique, which is universally adapted, it would have taken us many years to use 1,200 gene mutants to inoculate on this. So we used a new tool, new in vitro technique where we had this type of that is TIVIC technique. We immersed our TIVICs in a Hogland solution and inoculated, and it gave us results similar to that. And we, after inoculating these 1,200 mutants, now these are random mutants, so the tDNA might have gone into different genomes and some of the virulence genes, we had 2% genome, 2% the mutants, which were unable to infect this susceptible cultivar. So we have this now with the mutants which are going on, and their genomes are being, their genes are being decoded. Now coming to the R genes, as we know that the resistance genes, incorporation of resistance genes into a susceptible variety is very, very economic, because the farmers don't have to rely on the inputs like pesticide inputs on management of a disease. Now, over more than 40 genes have been identified till date, and only 17 of them have a number. Now, there is a number. This number is given by Bean Improvement Cooperative in USA. We call it as BIG. So there is a genetic committee which names them, and these are the well-named genes along with their chromosome number and that. Now, if you look at this, there are 4 genes which, using the test of allelism, show that they are totally different genes. But since they are on the chromosomes, which where already the number genes are, so it's very, very important for these authors to convince this BIC about this. And only in Co17, there is a second gene, Co42. It's a very, very important gene in G233. As I told you, there is only hardly any isolate which is able to overcome this resistance in this. And this is the gene which is actually responsible for it. Now, there are some genes which are tightly linked. For instance, we have a CO1 locus, which is at physical position of 50.229 MB at PV01, that's chromosome 1, and it's tightly linked with phase 1. They co-segregate, they inherit together. Similarly, you have a rust resistance gene, angular leaf spot gene, and a CO3 anthracnose resistance gene, which are inherited together when we work on these. So these are the genes which are distributed across all the chromosomes except 5, 9, and 10. So these 3 chromosomes doesn't contain any anthracnose resistance genes, even though recent study shows that there are some of the NBS-LLR genes on these chromosomes. I'll be coming to that also. Now, we did, since biparental population is very, very difficult. So Indian diversity panel, we had an Indian diversity panel with 230 entries. We inoculated them with 8 different races. This is the resistance scenario. Now, this race 2047, I will be highlighting this because it is one of the world's dreaded race around the world, even though 3481 is more than, but it is a dreaded race. Only 8 genotypes we found resistant to it in this. And when we did our genome-wide association using SNP markers, we found that the chromosome 1 contains our significant SNP, and when we decoded this SNP, we reached to a conclusion that this gene, it's a gene, a candidate gene, and it encodes leucine repeat, which is a typical signature of our gene. And we reached to this gene also. When we came to the race 39 and 55, we picked up a significant SNP on chromosome 4, and it belonged to a different novel region. Now, similarly here for race 7 and 10, we picked CO3, which is at PV04. It's a universal gene. It has many genes, and we also picked a CO2 gene, which is located to chromosome 11 in this. But when we did a QQ and our Manhattan plots were wavered, as far as race 2047, we could not pick any significant SNP. You can see how our QQ plots and Manhattan plots are wavered. And we mapped this gene, previous gene, which we identified on PV01. This is its genetic map, physical map, and we mapped it with SNPs, as well as we developed an indel marker, which is co-segregating with our CO1 locus, and it's 0.1 centimeter, very close to the gene, and it's being exploited worldwide now by breeders to integrate this gene into the commercial varieties. Now, transcriptome profiling, as I told you that earlier, it was not known how this resistance in this pathosystem is governed. So we had a nil pair, near isogenic line pair with CO1 locus, same with. So we inoculated this with array 73. We took the samples at different stages, and we had a high quality reads, which we mapped on the Phaseolus vulgaris genome, high quality our scatter plot as well as volcano plot show differential gene expression, many genes which are being up and down regulated. You can observe, you can see it here. So after analyzing, we found that at 0, there was no gene which was expressing, as it's obvious, but at 24 hours, the susceptible genotype, which was lacking the CO1 locus, the expression of the number of genes was higher in this case. But as soon as 72, this, as I told you, that this host pathogen interaction is an hemibiotropic type, and 72 is a very, very crucial. It is a biotropic nectrotropic civic, where the plant decides whether I have to go towards nectrotropy or I have to resist. Here we identified majority of the genes differentially expressing in resistant plant rather than in the susceptible. But later on, and when we did the downstream analysis and when we did for Go gene ontology, and we identified that most of these genes are sugar transporters. And these sugar transporters are very, very important for infection as well as living of this host bean pathogen on the bean. And same thing is happening at nectrotropic stage. And we had identified some of the major genes like CO4 and CO1, and we identified these of the say 6 genes which are differentially expressing at different fold chain levels. And there was an interesting, when we were doing the same study, when we published it there, we came to 6 candidate genes at CO1 locus. But within 2 years of our study, what happened that Chan et al. Identified a same gene on same chromosome with same location and named it CoYY. And earlier, the French group had already identified CoX gene on same chromosome, and it was using a test of allelism. It was shown that it's a totally different gene than CO1. So and they fine mapped these genes, we got the candidate gene list from those papers. When we aligned, when we mapped those streets, we were interested to identify that all these reports have 4 candidate genes in common. So we took the advantage, used the real-time expression analysis of these genes over a time course, and we identified a candidate gene that is FULVAL 800, which is a leucine-rich repeat gene for this interaction. And now it's being validated that it is actually a typical gene, which is actually imparting a resistance to anthracnose to wide different races of the pathogen. Now, as I told you that the bean anthracnose is highly diverse, being common bean is also highly diverse in northwestern Himalayan region. It was lacking that whether our beans, which are grown by the farming community of northern India, does they belong to Indian or Mesoamerican gene pool? So we sequenced a whole genome sequencing of 188 genotypes. We had 4 controls as well, with 2 typical Indian and 2 typical Mesoamerican genotypes. So we resequenced, we then identified nearly 25,000 SNPs. And when we analyzed, we were surprised to see that we had a typical Indian genotypes as well as the Mesoamerican, the 40 to 60% ratio. And we had some admixtures also. Here also, you can check it here also, here also. So this admixture is very well known in beans as well, but our beans, which are grown in the Northwestern, does belong to the 2 major gene pools. So we used the genome-wide association study here also. When we did the SNP analysis with race3, we picked the chromosome 4, and it was typical to CO3 locus. And when we used race 73, we picked the PV8, which contains CO4 gene. Now, this is very, very important. As I told you that our G233, which is a very resistant variety, doesn't flower and doesn't adapt to our environments. But our material, which we had a whole genome sequenced and analyzed it, and we identified this gene, very, very close SNP markers to that gene. And this is very, very interesting. And our breeders can take the message that our cultivars does contain this CO4 gene, and it can be easily exploited and introgressed into, because these are adapted to our environment, and we can easily introgress them into our cultivars, susceptible cultivars. This is, again, we picked it with when we did race 503, again PV04, and this is our most of the genes which we had significant genes are the typical resistance genes, and there are some significant SNPs which are out of the on studies which have not been unique SNPs were identified, which gives us an indication that the common beans grown in Northwestern may contain a typical different resistance genes. Now I'll be coming to one of the typical genes, like this KRC5. We studied it since 2006, and we found it resistant to more than 17 races. And we made developed a real population, F2 population. When we phenotyped this F2 population, we found the two races, race 3 and race 211. We found that there is a 3:1 ratio showing typical dominance of single resistance gene in both these. And we find when we mapped, when we inoculated phenotype our real population, it was segregating 1:1, giving a classical genetic analysis. But to our surprise, when we inoculated these real lines separately with this, we found a contradiction that some varieties which are resistant to race 3, they were susceptible to race 211. And it was inverse. So we hypothesized that there must be the two genes. It's not a single gene in KRC5, it should be the two genes, which we hypothesized this. Now we took the advantage, and using this, we did a BSA-sec, and to our surprise, we reached to the conclusion, yes, there are two independent dominant genes in KRC5, which our genetic analysis was not able to distinguish because the races were different. So it is that PV0, CO4 and CO10. As I told you that PV10 chromosome 10 doesn't contain any resistance gene. No resistance gene has been reported till date on this chromosome. So we identified a novel gene in this, and after browsing the genome, we could find that our chromosome 4 had 4 candidate genes which are having a typical resistance gene signatures. Only one of them can be a resistance gene for race 3 or race 211. Now here, when we did a PV10, that's chromosome 10, when we browsed the genome, we found multiple genes, nearly 20 genes are there, which are having an NBS ARC domain and LLR domains, typical resistant gene signatures in it. So this region is rich in these genes, and any one of them will be the candidate. We developed the indel and SSR markers. We mapped it. We mapped it with SCAR marker and indel markers with 0.8 centimorgan. And this work is under process because our student is currently working and fine mapping this gene on it and identifying the candidate, which probable candidate it will be. So since we don't have any map for this, so we did a DD-RAD-sec, and for the first time developed a linkage map, SNP-based linkage map for this common bean in India. Now I'll be coming to how these studies will help us in understanding the future dialogue. As I told you that we had already developed a repository, and our student is using tail PCR, and one of our students is doing CRISPR-Cas genome editing to identify these genes and their functionality using what the already ATMT protocol has given us, the repository. And this future prospects, as this paper recently came in 2022 in European Journal, that DCROTS, again, that same group, used our ATMT protocol and could identify homologous recombination, but with the difficulty. So we have switched it to the CRISPR-Cas based system, exploring this paper's the repository, the genes from this paper, so that we could easily functionally validate these genes and identify which genes are expressing at biotropic and then biotropic necrotropic switch. So our effort is on this. Now we have our student is also working on Northwestern Himalayan population using the SSR markers. As I told you that CO1 is an atypical resistance gene. So studies must be undertaken to knock down this CO1 gene, because a lot of confusion at this. So many genes are reported and all are naming them differently. But we are hypothesizing that there is a CO1 locus, not there. They must be allelic to CO1. They must not be a different gene, because they are residing all on just 100, not more than 0.000 kbs, just few hundred base pairs apart from each other. So this is what we hypothesize, and as our genome-wide association studies already identified novel genomic regions for our races, and those genes are need to be mapped and using the cutting-edge research tools. And as we identify the CO4 gene in our genetic material, it's now very easy to integrate this in using marker-assisted breeding approach. Now our system biology is needed. We know that Phaseolus vulgaris genome is already available. Polytotrichum dendrimicianum genome as well as its repository is available. Now we have some bioinformatics tools where we can use the effector proteins from this and NBS-LLR genes, which are now very well updated. Now we can use these interaction, that interlog-based interaction, for identifying these resistance genes. So I must acknowledge the research fellows, the SCOST, the HPKB Palanpur, then the funding agencies. I got the funding from SERB, DBT. I'm highly thankful to this UGC and CSI for funding me throughout my career for achieving these research goals. Thank you.
Yeah, thank you very much, Dr. Bilal, for the outstanding lecture today on the Phaseolus vulgaris collateral trichum lindenothianum interface inside and futuristic roadmap. Thank you very much, sir. You have to, I think, cover whole basics to applied and the management by the transcription projections and gene adapting. A lot of publication is published by you in reputed journals and by your team, sir. And I think you have managed the disease by identifying the various genes responsible for the disease, especially in the to manage the Rajma and Chawal in the Kashmir, Jammu and Kashmir. And you are basically provide all the information related to the first area of the collateral trichum to the future perspectives. Thank you very much, Dr. Bilal, for wonderful outstanding lectures, vibrant lectures. It will serve to our participants and member of the society, students working in the field of research and management of the plant disease, especially in the collateral trichum, sir, and also its extension workers, policy makers who have directly or indirectly involved in the collateral in our country for the planning of this disease. Vibrant lecture, sir, on this topic. Now I invite to our participants for the open discussions or questions. Sir, Dr. Bilal, participants, members. Dr. Prasad, if you have any queries, sir.
Dr. Bilal, thank you for such a comprehensive presentation and comprehensive, very nice work which you have done on the collateral trichum lindenothianum. This variability study starts with the in a crop, if a particular disease is being caused by a particular pathogen, and we look for availability of differential races to assess, to start with. Are you hearing? Can you hear me?
Yeah, I'm hearing.
Just a wild question is like if the crop having a particular pathogen which is causing a disease, and if against that disease in a crop, if differentials are not available, how one will go ahead to assess the variability of that pathogen against the particular crop?
Yeah. But there are so many host pathogen interactions where you don't have an international differential set. For instance, take an example of your chili anthracnose and chili genotypes. We don't have an international differential set there. But what you have, if you have a germplasm, you can use few of the isolates and look for the differential interaction, because you should have a differential interaction there. You can make your own differential set, and you can't categorize them into the races, but you can easily categorize them into the pathotypes. But unless and until there is not an international differential set available. So the results are very, very difficult to compare. Because you will be using a different differential set. Somebody else will be using a different differential set. It is very, very tedious at that time to compare the results.
Yes. That will question the validity of the results, even if the differentials are brought, isn't it?
Yeah.
Thank you.
Any more
Dr. Amjad
questions?
Hussaini, can you hear me? Dr. Amjad Hussaini, are you there?
Yeah, he is there.
He's there. Busy.
Any other questions by the other participants or members? Sridhar sir.
Yeah, very, very impressive talk on identification of the resistance genes, the pathogen biology, and variations, and other things. I have been associated with it for a long time. Certain things which occur to me is regarding the previous question, follow up on the previous question about differentials. It is not necessary that you should wait for the international differentials to come. Since you have the genotypes and you have also attempted to develop one near isogenic line, I think, in one of the slides which you've shown. It's possible for us to have an Indian differential set and then keep on identifying the pathotypes or haplotypes, you may call it as an haplotype even, which are varying in their virulence. And then probably I think this information will be useful for ascertaining the usefulness of the genes for deploying for resistance by the breeders. That is one. And I think since you have the know-how for developing isogenic lines, maybe perhaps you can put the genes which are available with you, at least the selected ones which are very promising and which are also succumb to the disease in certain areas, both combined together the genotype, and then put them on near isogenic lines so that you can develop your own differentials. And maybe I think others will follow when you develop your differentials to use for them. That is what occurred to me. But on the whole, I presume that the overall approach of your investigation relies on depending upon a single major genes for deployment, so that you can have a full level of resistance. But since you yourself have realized that the pathogen is variable, our experience not only with rice but also with the other crops have shown that when you deploy single genes, you are giving an opportunity for the pathogen to mutate and then overcome that resistance with aggressiveness, more aggressiveness, probable mutations. Probably, I think, since you're also familiar with the marker resistance selection, which you mentioned in one of your slides, you may try to probably put more than one resistant gene in the genotypes, so that the breakdown of resistance is a little bit difficult for the pathogen. Thank you. You have comments on that?
Thank you, Sridhar sir, for giving this. Yeah, it's very, as I told you, that nil pairs, as far as nil pairs are concerned, now developing the nil pair in a genetic background, having a universal susceptible, we should have a universal susceptible for that. Now, when this differential set was developed by Pasteur Correls, he intentionally used a Michelite cultivar and named it as a universal differential set. But within few years, and there were isolates which were unable to overcome the resistance. So later on, in the new gene, we have to identify first a genetic background which should be a universally susceptible to develop this near isogenic lines very well. It's very, very well known. And out of these common beans, we have one variety which is universally called as bread hog. Its name is bread hog. It's a black Mesoamerican variety, and it is universally susceptible to many of the isolates. But still, we have to wait. We are inoculating this so that we could develop a nil pair having a resistant, a single gene, so that we could categorize these isolates, as you rightly said, into haplotypes rather than on races, so that we could. And the marker-assisted breeding is going on. We have the varieties with the COFO gene now.
Thank you. My congratulations to you for a very good piece of work. Thank you.
I think Dr. Bilal is presenting very well presentations, and I think answer is also there in your presentation. There is no more questions. No doubt, sir. And now I invite to our President, Dr. Pratibha Sharma, Madam, Indian Phytopathology Society, New Delhi, for the vote of thanks to Dr. Bilal and our participants. Well.
Thank you, Dr. Pramod. So, indeed, today's lecture was worth listening, and I'm highly thankful to Dr. Bilal, who has agreed and really delivered his lecture within, I think it was just a short time request, and he has delivered. Monday we made the request, and Wednesday he is here. It was absolutely, like I felt I was listening to a lecture of molecular plant pathology, basics of plant pathology actually. So it was host pathogen interaction. You have very nicely worked. Actually, when Pramod was saying that Rajma, like we only know it is Rajma, and we are just having a very good dish out of this, but it had such a wonderful background of plant pathology and very well explained by Dr. Bilal. You have really worked a lot on this subject. So we are really thankful to all of you, all the audience, actually, the whole participants, and thankful from Indian Phytopathology as well as from myself. Another word that I want to request Dr. Bilal, that kindly please send your detailed this biodata also to Indian Phytopathological Society. While I was listening to you, I was trying to search your biodata and I could not get it. So please, please send us. You're a very important component of Indian Phytopathological Society, working in such a, a subject which is presently, I know students, especially for students, this lecture was really worth listening. They should listen that how pathogenic diversity, and then you have from the races, and then resistant genes, and then you have mixed up, then you came to the different races. So very well planned, and where the experiments are going on very nicely. And I think your work is, you have also given us a future roadmap also on this work, on this particular disease, which we really considered always in the field. I see the symptoms, but we were not really going into deep of this, this type of subject. So thank you very much, Dr. Bilal. And thanks to all our participants for being participating. And I thank my coordinating team, especially Dr. Gupta, Dr. Prashant, Dr. Prakash, who are always in the forefront and helping us out to conduct this Phytopet talk series. Otherwise, and it's always coming in time. Thank you very much.
Thank you, ma'am. Thank you, ma'am. Thank you, ma'am. Thank you, Dr. Bilal.
Thank you.
Sir.
Again, Dr. Bilal.
Thank you. Thank you.
Okay.